Welcome to the fascinating field of microbiology. In this course, we will delve into the intricate world of infectious agents and explore the invaluable role that molecular techniques play in understanding, detecting, and combating various diseases. We will examine the cutting-edge field of genomics and its applications in epidemiology, antimicrobial resistance, zoonotic diseases, outbreak investigations, vaccine development, host-pathogen interactions, surveillance systems, metagenomics, molecular diagnostics, and epidemiological modeling. Phylogenetics is the study of the evolutionary relationships among organisms. In the context of infectious agents, it involves analyzing their genetic sequences to determine their relatedness and evolutionary history. This is done through techniques such as phylogenetic trees, which depict the branching patterns of pathogens based on their genetic similarities and differences. By constructing phylogenetic trees, we can trace the ancestry of pathogens, identify their sources, and uncover patterns of transmission. We can also gain insights into the evolution of virulence factors, drug resistance genes, and other important genetic traits. Phylogenetic analysis is particularly useful in tracking outbreaks and understanding the ways in which infectious agents adapt to their environment. Genomic epidemiology combines the principles of genomics and epidemiology to investigate the genetic factors influencing disease transmission and virulence. By sequencing the genomes of pathogens, we can identify genetic variations that contribute to their ability to spread and cause severe disease. Understanding the genetic determinants of transmission can aid in the development of targeted interventions. By identifying specific genetic markers associated with transmission, scientists can track the movement of pathogens and implement effective control measures. Additionally, genomic epidemiology can shed light on the molecular mechanisms underlying pathogen virulence and guide the development of novel therapeutic strategies. Pathogen typing classifies strains based on genetics, using methods like MLST, PFGE, and whole genome sequencing. MLST sequences specific gene regions, PFGE separates DNA fragments by size, and whole genome sequencing provides a comprehensive view. These techniques help track pathogen spread and transmission during outbreaks. In summary, studying the molecular aspects of antimicrobial resistance helps us understand how resistance develops and spreads among pathogens. This knowledge is vital for developing strategies to combat resistance and improve treatment options in medicine. Zoonotic diseases can jump from animals to humans, and understanding how this happens at a genetic level is crucial for controlling these diseases. By studying the genetic changes in pathogens, we can learn how to prevent future outbreaks and develop vaccines and treatments. During disease outbreaks, it is important to quickly and accurately identify the source, reservoir, and transmission pathways. Molecular tools like genome sequencing help us understand the genetic relatedness and transmission dynamics of pathogens, aiding in outbreak control and prevention strategies. Vaccination is vital in preventing diseases. Molecular methods help assess vaccination impact and efficacy. Monitoring pathogen changes helps update vaccines against emerging strains. Molecular techniques aid in vaccine development and improve efficacy. To study host pathogen interactions, we examine genetic factors that influence disease outcomes. Molecular studies help us understand how host susceptibility and pathogen virulence are determined, leading to targeted interventions and personalized treatments. Analyzing host and pathogen genomes identifies specific genetic variations that impact susceptibility. Additionally, understanding pathogen virulence at a molecular level allows for the development of therapeutics that target key genetic factors, potentially reducing the impact of infectious diseases. Surveillance systems play a crucial role in monitoring the emergence and spread of infectious diseases. Molecular surveillance methods provide a powerful tool for tracking pathogens and understanding their population dynamics. By deploying molecular techniques, such as real-time PCR or high-throughput sequencing, we can detect and identify specific genetic markers associated with pathogen species or strains. This allows us to monitor changes in pathogen populations over time and across geographic regions. Molecular surveillance data can inform public health interventions, such as the implementation of targeted control measures or the development of new vaccines. By understanding how pathogens evolve and spread, we can better predict and respond to disease outbreaks, effectively reducing their impact on public health. Metagenomics is a powerful technique that involves sequencing the genetic material obtained directly from environmental or clinical samples. It allows us to study the diversity and function of microorganisms and identify novel pathogens. By analyzing the genetic makeup of microbial populations, we can gain insights into the role of the microbiome in health and disease. In summary, molecular diagnostics, including PCR and DNA sequencing, have transformed clinical microbiology by enabling rapid and accurate detection and identification of pathogens in clinical samples. These techniques offer faster results, increased sensitivity, and improved patient care and infection control. Molecular epidemiology combines genetic and epidemiological data to predict the spread and impact of infectious diseases. By analyzing pathogen genetics, we can track movement, identify high-risk areas, and inform interventions. It also helps assess the effectiveness of control measures. In conclusion, molecular techniques have revolutionized microbiology, providing insights into disease transmission and evolution. Understanding genomics, antimicrobial resistance, and outbreak investigations is crucial in improving global health. As healthcare professionals, we must embrace and apply molecular biology in our practice.